something happened. Okay, so I hope you will have again a better understanding of the research environment of the graduate school of pharmaceutical sciences. Okay. Um, okay, one piece of information that was not included in the short video is our department recently constructed a new building named Building 4, which is used for lectures and research. Actually, my laboratory will move to this building maybe next year. Okay. Okay, so now let's get into the main topics. So to begin with, since many of the attendees today are undergraduate students, so I'd like to explain the research area in the field of natural product chemistry. Okay, so in the area of natural product chemistry research, there are broadly 4 areas of study. It's blue color. So first one is traditional natural product chemistry research. So this research, as you know, involves collecting natural resources, evaluating the biological activity of their extract, isolating and purifying the secondary metabolite from those extracts, and determine their chemical structure. So additionally, 3 other studies revolved around the utilization of isolated bioactive natural product or extract of natural resources. So one involved total synthesis and analog synthesis, which eventually lead to the drug development. So another study investigates the mechanism of action and target molecule of isolated bioactive compound known as a chemical biology research. So this study contributed to the discovery of new drug targets and new metabolic pathway in the body. The last one is biosynthesis research, which for instance, in the case of microbial secondary metabolite, investigates the enzymatic reaction responsible for producing the compound within the microorganisms. So this type of research can discover the new enzyme or enzymatic reaction. Moreover, this research has recently expanded to heterologous expression of biosynthetic gene to produce secondary metabolite. So as now you may understand, in the area of natural product chemistry, these 4 studies do not exist individually. So to advance this area, it's crucial to explore new natural resource and discover new secondary metabolite, thereby revitalizing the entire field. Okay. So furthermore, it's hoped that in the future, alongside these 4 studies, a 5th new study will emerge. Okay. So in our laboratory, we conducted the traditional natural product chemistry research and total synthesis and the analog synthesis and the chemical biology research, all viewed from the perspective of natural product drug discovery. Okay. So this slide summarizes the study in my laboratory. So today, I'd like to present our research result in our screening resource and traditional natural product chemistry research and the synthesis and chemical biology research, including the latest research techniques. Okay. So let's start with the screening source, natural resources. So as you know, the study of natural product chemistry developed from interest in the medicinal effect and the constituent of plant extract. For example, poppy's extract has long been known to have anti-pain property. So research into the constituent of the poppy led to the isolation of morphine in 1806, and its structure was elucidated in 1952. And ephedrine was isolated in 1885, and its chemical structure was clarified in 1892. So even today, the analysis of the constituent of medicinal plant is one of the most important fields of natural product chemistry. And much research is being conducted in this field. So in other words, plants are still a good natural resource to isolate bioactive secondary metabolite. So subsequently, the accidental discovery by Fleming that fungi produce antimicrobial substance, penicillin, and the discovery of streptomycin from actinomycetes by Waxman led to the focus on microorganisms as a medicinal resource. So these discovery also established the concept of actively searching for medicinal seeds. And many researchers and pharmaceutical companies began to develop the search the medicinal seeds. Okay. So, return to natural resources. Following terrestrial medicinal plant and terrestrial microorganisms, organisms and marine microorganisms have attracted attention as natural medicinal resource. So this slide shows why researchers focus on marine medicinal resource. So as you know, 80% of marine organisms and marine microorganisms are said to be undeveloped. They are living in 3.4% salt water. They are also affected by water pressure, and sunlight is difficult to transmit as compared to land. So from these, marine organisms and marine microorganisms are considered to have different metabolic pathway compared with terrestrial microorganisms and plants. So in other words, it is expected they have the ability to produce different new compounds from terrestrial microorganisms and plants. So many researchers are conducting the research focusing on the marine medicinal resource. So indeed, several compounds and their derivatives from marine have already been clinically used. For example, anti-cancer drug, trabectedin, DNA chelating agent isolated from tunicate. So therapeutic agent for pain named ziconotide, inhibitor of N-type calcium channel, isolated from shellfish. And one more example, anti-cancer agent, eribulin, inhibitor of tubulin polymerization, was developed using yeast parts of Halichondrin B isolated from marine sponge. And more than 20 marine-derived compounds or their synthetic derivatives are still in clinical trial. Okay, so, and I'm also working with Professor Andy Setiawa and Professor John Henry to search for bioactive natural product by collecting marine organisms and marine microorganisms at the location shown here. So we have also published a number of research results as a collaboration, and I'm very grateful Professor Setiawa and Henry for their collaboration. So I hope to continue the collaboration with them. Okay. And also, this is a message to the student and the young researcher based on my experience regarding collecting marine natural resource in the Southeast Asia. So actually, Indonesia's natural resources are more wonderful than you can. So please value Indonesian natural resources and create new way of utilization of them. Okay. Okay, so now I'll showcase through a short video how we conduct the collection of marine organisms. So here is around 15-meter depths of Kupang in Indonesia, as you know. As you can see, they have very wide diversity, and the colony of sponge are quite large. So we always dive and collected marine organisms, yes, like this. And we collect one species from 200 to 500 grams, and number of sample is about 30 species in one trip. So then we also isolate marine-derived microorganisms from collected sponge and sea sand. And these organisms collected in this manner undergo extraction process, resulting in preparation of extract library. In addition, isolated microorganisms allow for the creation of culture extract library. And we also perform the LCMS analysis on this library. Okay, so I skip this. Okay, so subsequently, we will conduct the screening to find bioactive extract on the marine natural resource library. So I will now introduce our bioassay systems. So before introduction of bioassay, our bioassay system, so the method for evaluating biological activity include phenotypic screening and target-based screening. The phenotypic screening use the cell, microorganisms, or animal to measure the activity of sample based on its phenotypic change. On the other hand, target-based screening basically use enzyme and other substance. So typical example of phenotypic screening include cytotoxic activity test using cancer cell and antimicrobial activity test using pathogenic microorganisms. On the other hand, target-based screening include antioxidant activity test and phosphatase inhibition test. And each has its advantage and disadvantage. So specifically, phenotypic screening is likely to discover active natural product with novel mechanism of action. But it's developed a compound as a drug, its mechanism of action and target molecule must be clarified. Okay, target-based screening, on the other hand, do not require clarification of the mechanism of action or target molecule. But the compound may not show the activity in cellular level or animal level. Okay, so in my laboratory, we mainly use the phenotypic screening system to search medicinal seeds, mainly because we are interested in the compound with new mechanism of action. Okay. So this slide shows our recent phenotypic screening system. The first is screening for anti-cancer agent. And the cancer cell adapted to a low nutrient environment, which are partially present in tumor, are considered to be a major cause of pathology of deterioration. So we are searching for selective growth inhibitor against cancer cells that are adapted to a lower nutrient environment. And this is a screening system searching for a compound that inhibit cancer metastasis. So as you may know, exosome or we can call extracellular vesicle, EV, which are nanoparticles secreted by cancer cell are involved in cancer metastasis. So we are searching for a compound that inhibit the exosome production from cancer cell. And we are also interested in infectious diseases. So for example, dormant states of Mycobacterium tuberculosis caused by environmental stress at the infected site is a major therapeutic problem because it's resistant to anti-TB drugs. So we are searching for antimicrobial substance that are effective against the dormant Mycobacterium tuberculosis. In intermicrobial communication and are searching for the compounds that disrupt the interaction between intestinal bacteria and amoeba, thereby inhibiting the growth of amoeba. Okay. So in addition, a number of compounds have been found in the past few years, especially from the screening system for exosome production inhibitor. So one of these is this one, asterotoxin, isolated from the culture extract of Aspergillus genus, isolated from a marine sponge. So this compound inhibits exosome production with IC50 value of 0.5 micromolar. So additionally, it was indicated this compound inhibits exosome secretion by promoting the degradation of exosome within the cell. So another compound was also discovered from the culture extract of other Aspergillus genus of marine origin, which present as a dimeric compound. So upon brief consideration of structure-activity relationship, it was suggested that a dimeric structure is necessary for showing the activity because the monomer did not exhibit any activity. So additionally, due to the processing D displays the most potent activity. So we believe that the presence of G-chromen structure and this hydrox group are crucial for the activity. So currently we are progressing with the analysis of mechanism of action and the target molecule of this compound. So furthermore, really interesting things is during the screening of substances that inhibit the exosomes production, we found the presence of extract that increase exosome production. So we contemplated utilization such extract as a cosmetic ingredient. So specifically, extracellular vesicle exosome produced from adipocyte derived mesenchymal stem cells, we can call ADMSC, are known to be taking up dermal fibroblast. And then the dermal fibroblast increase the production of collagen and elastin. So compound that enhance the exosome production from ADMSC were considered as a cosmetic ingredient with anti-aging effect. So in this case, although extract was from plant origin, but eventually we identified these oleanan type triterpene saponin as the active substance. So additionally, the structure activity relationship indicated the having here. So having 2 sugar at the third position is better than 1, and uronic acid is preferred for 3 position. And indeed, we have found that culture supernatant of ADMSC treated with this saponin enhance the collagen production in fibroblast cell compared to untreated culture supernatant. So we are currently conduct the research on this compound in collaboration with a company to develop as a cosmetic things. And beside this, we have so far found bioactive compound as shown here. So my laboratory also have a synthesis group which conduct research on the total synthesis of these compounds and the synthesis of analog compound with the aim of developing them as medicinal seeds. Okay, so by the way, structure determination of natural product is generally performed using mass spectrometry and NMR. The sensitivity of NMR has improved, really improved actually. However, it is still desirable to isolate a few milligrams of compound, especially for compound with complex structure. On the other hand, 2 structure analysis methods using X-ray crystallography have recently attracted much attention. One is a crystal sponge method using a porous complex developed by Professor Fujita at the University of Tokyo. So this is a method of X-ray crystallography after introducing the compound, even oil form, into the porous complex, and it's possible to determine the structure of compound. So they have also successfully determined a few micrograms of compound that they have separated by HPLC. So the other method utilize the electron microscopy, which has been used successfully to determine the structure in a single piece of nanocrystal. So these techniques are very useful in determining the structure of natural product with a trace compound and will be actively used in the field of natural product chemistry in the near future. And Also, I have started the collaboration with collaborate with Dr. Fujita using the uh, porous complex. And also, really lucky things is last year the Japanese government provide the cryo-electron microscopy to the Osaka University. So now we can start to use this machine to elucidate chemical structures. And next, I'd like to introduce synthetic research and chemical biology research. So now in our laboratory, we are conducting the target identification study of isolated bioactive compound using these methods, such as analysis of gene expression profile, the method using the transformant library, and pull-down assay using synthetic probe of isolated compound. And so far, we identify the target molecule of these compounds, as you can see here. So today, I will briefly present 3 research examples. Okay, so target identification of a compound called radiacin A identified as an effective antimicrobial substance against the dormant bacteria is the first example. So this compound, a unique cyclic peptide with a raster structure, was discovered from the culture extract genus Rhodococcus. So we conduct the target identification of this compound using Mycobacterium smegmatis transformed with genomic DNA library prepared from genome of Mycobacterium species. So specifically, we screen for Mycobacterium smegmatis transformed with the genomic DNA library that shows a resistance to the radiacin A. So eventually, we investigate which gene from Mycobacterium BCG, when overexpressed, confer the resistance to radiacin A. So as a result, it was found that overexpression of repressor INBR or delta subunit of ATP synthase give resistance against radiacin A. So consequently, we currently consider the target molecule of radiacin A to be INBR and the delta subunit of ATP synthase. So this is target identification of PDOA, another antimicrobial compound effective against dormant mycobacteria. So the identification of target molecule of PDOA has not been achieved yet, actually. But I will introduce on the process made thus so far. Okay, so this compound isolated from Indonesian marine sponge of Arctos species, and this compound not only shows a potent antimicrobial activity, but also proved effective against multi-drug resistance of Mycobacterium tuberculosis. And the successful total synthesis has been accomplished. Subsequently, we conducted structure-activity relationship study using synthetic analogs. So the result indicated the conversion of R1 position does not impact the activity. However, modification in R2 lead to reduce the activity. So based on this structure-activity relationship, we designed and synthesized probe incorporating diazirin group for covalent binding upon UV irradiation, as well as here alkyl moiety enabling biotin tag introduction. Okay, so currently, as a strategy, we mixed the probe with bacterial cell lysate, perform UV irradiation to form a covalent bond between the probe and the target protein. So subsequently, introduce the biotin tag via using a click chemistry, and finally precipitate the probe target protein complex using a structural beads for identification of target molecule using LC mass. So while I will only mention the outcome, so we currently consider these four bands presented here as a candidate target molecule and are conducting LC mass analysis for identification. Maybe I hope next week I can understand we can understand these protein. Okay. So the last example involves the target identification of Orianan type triterpene saponin, as mentioned earlier, which stimulate exosome production. So for this purpose, we synthesize probe molecule as shown here. Then we examine whether or not the compound could enter the cell. So typically, the saponin are believed not to enter the cell. However, our preliminary result suggested the compound might enter the cell to some extent. So currently, we are proceeding with the research under the assumption that target of compound might be intracellular. Okay. So these introduced studies are very important in drug discovery research. On the other hand, it also true that such a study are time-consuming and expensive. But recently, a method called network pharmacology, which predicts the mechanism of action of compound or extract and its target molecule, is being established by utilizing a gene card and the cake pathway database. And in the field of the drug, crude drug, TCMSP database and the AI function. So if we can successfully incorporate this method and proceed with target molecule analysis of the isolated bioactive natural product, so we will be able to identify the binding protein of the compound much more quickly than before. So I would like to establish the system to implement this method in the laboratory as soon as possible. Okay, so this slide shows the for research within the area of natural product chemistry that were initially explained. So as mentioned before, the initiation of new research is desired in the field of natural product chemistry. As parts of such research endeavor, we have started on studying intermicrobial communication. So through this research, we will have new bioactive natural products and gaining insight into why microorganisms produce secondary metabolites. So as you know, microorganisms have many dormant biosynthetic gene clusters that remain inactive under typical culture conditions. So it's expected activation of these dormant gene clusters could lead to discovery of novel secondary metabolites. So we are conducting the co-culture experiment involving Mycobacterium smegmatis and marine-derived fungi as a method to activate the dormant genes. So specifically, Mycobacterium smegmatis was used for activator of dormant biosynthetic gene cluster in fungi. So more specifically, 100 strain of marine-derived fungi was cultured with Mycobacterium smegmatis. So after that, we prepared ethyl acetate extract from each culture. Then the biological activity and compound profile of each extract were compared to the extract of fungi actinic culture. So as a result, co-culture extract of marine-derived fungi, Aspergillus niger, and the Mycobacterium smegmatis show the cytotoxic activity and production of pigment. In addition, the profile of secondary metabolite also changed significantly, and we found that compound A2G was selectively produced by co-cultivation. Then we identified the malformin C as a cytotoxic compound, which is selectively produced in co-culture. We also isolated TMC256A1 and desmethyl cotanine and orasperon C as a compound that was selectively produced by co-cultivation. So from these results, we believe that co-culture system using Mycobacterium smegmatis can improve the diversity of secondary metabolite in the extract. Okay, so next, two studies were conducted as a part of analysis of mechanism of action. So as a first experiment, we cultured Aspergillus niger with autoclave. It's dead to Mycobacterium smegmatis. So after that, we confirm whether the extract shows the cytotoxic activity or not. So as a result, the cytotoxic activity of extract was same as in the extract of actineric culture, you can see here. So this result indicated living Mycobacterium smegmatis is necessary to produce the cytotoxic compound from fungi. So next, as shown here, the Mycobacterium smegmatis and Aspergillus niger were cultured separately by using the dialysis membrane. So as a result, the cytotoxic activity of extract culture under this condition was the same as in the actineric culture condition. So these observations indicated Aspergillus niger needed to contact directly with living Mycobacterium smegmatis for producing a cytotoxic compound. So currently we are investigating which molecule of Mycobacterium smegmatis activate the production of cytotoxic compound. Also, we have planned to investigate which type of signal transduction pathway is activated in Aspergillus niger. Okay, so this is last slide, and today's lecture is a bit rambling, but we will continue the research to find new pharmaceuticals and new drug target incorporating new knowledge and technology. And I'd like to continue the collaboration research with Rampur University, Professor Andy Setiawan and John Henry. So thank you very much for your kindly attention.
Okay. Thank you very much, Ari Sensei, for your nice presentation and very interesting. And we now to start discussion session. So to all participants, if you have question, please you can direct to Ari Sensei or you can write your question in chat room. Any question?
Pak Saiful.
Ya.
Oh, Pak Heri dulu, silahkan.
Halo.
Please.
Okay. If it's okay, I start to discuss since we waiting for the student question, I think, because of our inbound lecturer is short for our student here. But I think in this program also, some of the lecturer attending this event. So if it is okay, I start to discuss. Okay. Thank you very much for Professor Masayoshi Arai for nice presentation. I think as a biochemist also, I have some difficulties to understand about the structure of the compound because of they are very complicated, and I think many various of the structure could be characterized. It is very nice work. So as a biochemist, I think I work also with the bacteria and also some of microorganism, uh, but uh, but I think not in this field. But and then for the pharmacist field, I think is it the compound that you already explained to us produce extra cellularly or intracellularly because of the last part of how the interaction between Aspergillus niger and I forget the other name of the microorganism is attract me that so they need to interact with one and each other directly. I think so. Uh, I guess because of we teach our student that some compound can produce extracellularly, and then some compound produce intracellularly, and then it is I think how this compound many compound produce in marine microorganism or marine bacteria. And then because of I think the student here that attending our class is a new student that have not already known about the organic chemistry structure. So there are many structure of the active compound that already explained to in our meeting now, and maybe would you please tell my student, or maybe I'm also, how we can find our attraction with the new of the organic compound structure that have good function in medicine or pharmacist field like that. So I think enough to of my question. Thank you very much. And for my student, we are also waiting for your question to give to our speaker now. Thank you very much. I'll back to Pak Saiful.
Okay, thank you very much to Mr. Satria. So, Ari Sensei, you can direct answer the question from Harry Satria, Dean of Faculty of Mathematics and Natural Sciences.
Okay, thank you very much for your comment and question. So, my understanding correct, maybe the if you find the new compound or if you want to find mechanism of action or new structure of compound, maybe the actually everything is important actually. Because one thing is source is really important, but Indonesia has a lot of good natural resources. Maybe you can use your natural resources directly to find new structure compound, or new compound has new biological activities. But same times, so we have to think about how to establish the screening system, how to evaluate biological activity. So this is also very important. So actually, if we screen just anti-cancer, for example, antimicrobial activity, it's difficult to find new compound. So that's why we have to change I have to change the strategy to find the antimicrobial compound. For example, we using the hypoxic condition to cultivate the pathogenic bacteria to induce dormant state. So this is one of the example. So we have to think about how to evaluate the compound. I mean, how to establish a new screening system. Is it okay for your question?
Yes, yes, okay. I can understand. Yeah. Yes, thank you very much, Prof. Mas Sayosi Arai. Yeah. Pak Saiful, your voice could not. Okay.
Okay, thank you very much to your question. And there is a question in our chat room. I will read the question from Siti Aisyah. Good afternoon, Prof. Arai. Permission to introduce is Aisyah, second year student in Master in Chemistry. Supervisor by Prof. Anistiawan. Please allow me to ask, what specific challenges or hurdles did you encounter during your experiments, and how did you address them? Thank you.
This meaning is maybe what is the meaning is
the specific challenges.
What do you mean in specific challenging?
Is it connected with some difficulties? Some difficulties that's faced by this research, like it, Siti Aisyah?
Okay.
Could you explain
Siti Aisyah, can you direct the question to Ari Sensei?
I guess it's Siti Aisyah who want to know about the challenges is like some difficulties that we face in to conduct this research. Siti Aisyah, oh, okay, please explain to Prof. Arai.
I'm sorry.
So, yeah.
You got specific challenges or hurdles, and how you fix it, and how you to deal with it. Thank you.
Not too clearly to attach your mention, Siti Aisyah. Maybe one more time, would you please to explain to us so Professor Aya can understand what are your question? Okay, please. One more time.
Mungkin di bahasa Indonesia dulu, Aisyah.
Halo, Siti Aisyah. Satu kali lagi dijelaskan di dalam bahasa Indonesia. We will help you. Sorry.
Okay, so I'm not sure this is for your answer or not, but the challenging things is so now I'm a professor actually, so there is a lot of students in my laboratory, and also there are students come from a lot of countries. So we have to manage of the student actually. So the level of student is different, and the character is also different. But basically, they are really good students. So my side try to encourage to each student. So this is a challenging one. And as a research, so basically we can isolate a lot of compound, but the difficulty point is development of this compound as a drugs. So we need a lot of experiment to develop the compound for the drug. So this is a challenging and difficult point in our research. Is it okay for you?
Yeah, it's okay. Thanks for answering me.
Okay, thank you very much. That's enough. So to all participants, ask your question in this room. Okay, Maulana Bintang. Ya, langsung.
Kamera dua dibuka, mic ini coba perhatikan. Kamera dua. Ya, silakan.
Okay, before I ask to you, I want to say thank you for your time in providing knowledge to us, to the Department of Chemistry, Faculty of Mathematics and Natural Science, University of Lampung. And my question is, after you have an active biocompound, what is the next plan for your bioactive compound? Synthesis or still extract them from the natural product? Thank you.
So your
Okay,
question
can you
is,
hear the question, Ari Sensei?
yeah, my understanding, his question is how to develop compound to the drug. Is it his question?
I think
Yes.
Maulana Bintang asked if the compound already obtained. So what is the next strategy? It is we are still using the bacteria originally or synthesized to produce this compound like that.
Okay, so from the aspect from the compound, we synthesize we synthesize analog compound because we have to increase the activity of compound and decrease the side effect. That's why we can try to synthesize analog compound. And from the viewpoint of the biological activity, so after using, for example, the pathogenic bacteria, so we can move to the animal level. So for example, so if you find the antimicrobial activity on the activity in the some extract or compound against the mycobacteria, so we will move to the infection test using a mice. And if the compound effective to mice, so we can move to the monkey, sometimes dog or something. So this is our strategy to develop the compound to the drug.
Okay, it's enough. Thank you very much. Your answer, Ari Sensei. And the next question from the participants. Any question? Okay, if you don't have question, so Mr. Mulyono, please, if you have question to Ari Sensei.
Okay, thank you for the chance to ask. I have three questions. First, in your laboratory, notice you focus to isolate a bioactive from fungi. Microorganism, of course, not from fungi, but also from bacteria and actinomycete. And then, what is the consideration? Recently, your laboratory focused on fungi. Actually, biar nggak tahu kalau sini.
Okay, so my interest is not only fungi. I'm interested in the actinomycetes, of course, also
course,
the
okay.
extract. But actually, the genome size of fungi is larger than bacteria. So that meaning is the gene cluster of secondary metabolite is much included in the fungi, I guess. That's why I'm recently I tried to isolate some compound from the fungi extract, but I of course interested in the actinomycetes and marine organisms.
Okay. Actually, after we meet last year, I contact with Mr. Andy group also to isolate from bacteria because here we have a limitation to overcome isolation from fungi. We focus to start from bacteria, especially from mangrove sediment, and some isolate also showed activity against the pathogen. So as you know that in here in our university, with the limitation of instrument, so what is your suggestion? The part of experiment that activity, for example, against the pathogen or having the other activities?
Okay, so basically, it's better to collaborate with Japan. And then also, my suggestion is, so maybe your university has a lot of type of pathogenic bacteria. So you can get a lot of pathogenic bacteria from the hospital. So maybe you can try to evaluate the antimicrobial, evaluate your compound against a lot of type of pathogenic bacteria from the clinical. And this is one of the suggestions from me. And also, if you elucidate chemical structure of compound, recently we are trying to establish the system to analyze the online system. So I mean, I'm trying to establishing actually. So if you send to your compound to the Osaka University, so we can measure the NMR, and then the data is transferred to your university using the online systems. So you can analyze yourself the data of NMR. So now our department tried to establish this system. So once we establish this system, you can analyze the NMR spectra yourself in Indonesia. So this is a future plan, maybe our collaboration.
Yeah, Thank you for your suggestion. I think it's very fruitful suggestion. So we are going to plan with Pak Andi and our group to more deeply prepare the examination of active compound that we can isolate from some bacteria, of course, from the marine environment. My third question is about your last presentation about the toxicity, cytotoxicity between Aspergillus niger and Sphegmatis. The autoclave Sphegmatis, is it kind of experiment of Avery and McLeod? So I mean, the part of gene will be combined with the Aspergillus niger, and then finally they can change the dormant gene function to be active. Maybe I lack this kind of information.
So I think maybe the surface of lipid molecule on the mycobacteria is stimulate the signal transduction of fungi. Maybe the no gene transfer between the Sphegmatis and the fungi. Just the surface molecule of Mycobacterium Sphegmatis stimulates the signal transduction of the fungi. This is my hypothesis.
Okay. It's very new techniques, I think, for us here. And for the network pharmacology, that kind of using GenCard and Kirkpatrick, it is kind of bioinformatics techniques to find this how to trial many function of the gene, human gene, and new drug. This kind, maybe, Professor?
Yes, that's correct.
Okay. I think we have to encourage a lot of more understanding this kind of new field that, of course, became very, very interested in know this. Okay, thank you, Pak Saiful, Prof. Arai.
Okay, thank you very much to Pak Mulyono of your question, and we have time to participants in this room. Please, if you have any question to our sensei, okay, stand up, please, and direct to our sensei your question.
Your presentation, we know that you already have so many research about anti-cancer compounds. Then my question is, what is the most obstacle in your research? Thank you.
It's difficult to hear important word. Could you repeat again your question?
My question is, based on your presentation, we know that you already have so many research about anti-cancer compounds. Then my question is, what is the most obstacle in your research? Thank you.
Okay, so maybe asking me about it's difficult. She asking me difficulty point.
May I have to repeat the question? She's asking about the after you find a lot of kind of bioactive compound. So what is the obstacles to overcome or to conduct the next research?
Okay. So is to develop the compound. I developed the compound for the drug together with a company. And so I'm interested in the mechanism. I want to create new drug using anti-cancer compound. So I'm thinking about two things. One is for drug and one is for creating the drug target.
Okay, thank you. The next. Okay, Fikri. Maju, maju, Fik.
Sorry, I can't hear you.
I have any question. My opinion question is, you have get sample in Indonesia. Do you feel Indonesia government support foreign research? Thank you for attention. You have get sample in Indonesia. Do you feel Indonesia government support foreign research? Thank you for attention.
Sorry, it's difficult to hear.
May I try to repeat? His question is about based on the lot of sample you had collected from Indonesia, what do you feel about government support for the research?
Oh, I see. So of course, the Indonesian support from the Indonesian government, also DP, now the name is changed, BRIN, is really important for the collecting Indonesian marine resources. So basically, we have a contract between the University Rampung and Osaka University. And also when I go to the Indonesia to collecting the natural resources, every time Professor Andy Setiawan and Professor John Henry help us to approve from the government. So, and also I'm thinking its important point is the data sharing. Of course, if we publish the paper, so as authors, the John Henry and the Professor Andy Setia were also including in the paper. And also, if we use some bacteria or fungi to something, every time I have to approve from the Professor Andy Setiawan. So it's important point is the communication of the collaborators, and also important to the government support. Okay.
Okay, thank you very much for your answer, Ari Sensei. And Fikri is enough. The other participants have any question? Okay.
Uh, I got a question. So, how big opportunity? How big the opportunity to from your potential compounds to as an anti-cancer drug? And is there any potential side effects from that compound? Thank you.
Okay. So I think the question is how much potential our compound has. Is it correct?
Yeah, potential compound applied for cancer. So you have already get the compound, and of course, are there any information about the side effect from the application of the compound?
Okay, so I'm trying to develop some compound for the drug with some company. Actually, it's really, really difficult to develop the drug. So honestly to say, the natural product cannot cannot use drug directly. So every time we have to synthesize analog compound using a basic natural product. But lucky things is, so now one compound or totally two compound from marine, the testing about toxic effect, the dog level. So if the compound is clear to cytotoxic test, maybe next step is human trial, but the problem is also the grant. So if we test if you test the compound in the clinical level, maybe we have to need more than 1 billion yen for one drug. So we have to think about how to get the grant to test clinical. But now we have several compound has a potential to drug.
Bisa ditangkap. Jadi untuk kesulitan aplikasinya masih pada level penerapannya masih pada di level anjing. Untuk ke manusia belum, karena butuh sekitar 10 million yen, right, Professor? Around 1 billion Indonesian rupiah.
1 billion.
Wah, 1 billion rupiah. Maybe Pak Andi and Prof. John Hendry can ask Ministry of Education. Yeah. Okay. Okay, bisa dipahami. Okay, the student already understood. There is one more student are going to ask.
yang dulunya pernah saya diberitahu juga bahwa dalam diri manusia itu sebenarnya terdapat senyawa kanker di seluruh tubuh manusia, hanya saja ada yang aktif dan tidak. Pertanyaan saya, apakah berdasarkan research dari Prof. Masayoshi Arai, apakah ada senyawa yang dapat secara langsung untuk mengaktifkan senyawa kanker tersebut di dalam tubuh manusia? Gitu? Apakah pernah meriset hal tersebut atau tidak?
I have to translate the question. He knows from doctors that in human body, actually there is a kind of a cancer cell. So is it right or not? Is it correct or not? One. And then other specific compound that come from outside that can repress this kind of cell that potentially to be cancer cell. Could
Sorry,
you catch?
your voice is still howling.
Suaranya. He is asking. He is asking about. Sorry, because there is a defect sound here. He is asking, is it correct that in a human body, there is kind of cell that tend to be cancer? Then, if there is a kind of cell that potentially to be cancer, so what kind of substance from outside that can repress this growth of cell and cannot stop or cannot to be cancer? She know this knowledge from doctors, medical doctors. So it is correct or not based on your experience?
It's a difficult question for me because I'm not medical doctor and my major is chemistry. So, but my understanding is the cancer cell coming from the aged cells, I guess. So actually I'm trying to find some compound to induce the death against aging cell specifically. So if we can remove the aging cell from the body, maybe the ratio of cancer will be decreased. Is it okay for against your question?
Gini, maksudnya beliau bukan seorang dokter bidang medis yang secara langsung mengetahui statement Anda tadi. Yang kemudian yang kedua, beliau melakukan riset tentang anti-aging, anti penuaan. Karena artinya sel itu ditreatment dengan substance yang diperoleh oleh grup beliau, terus kemudian kan jadi selnya tumbuh kembali itu. Namanya tumbuh kembali kan ada pertumbuhan di luar normal kan gitu. Nah, apakah itu kemudian juga potensial ke depan malah menjadi kanker, itu belum tahu. Ya. Kurang lebih seperti itu jawabannya. Bisa diterima? Oke, here already understood. Ada lagi yang lain? Another else? Oke, langsung.
Kamera satu di on. Oke, kurang.
From Professor Arai Research, is there any applied in pharmaceutical industry? is that applied in Japan or Indonesia?
The question is about from your compound that you collected or isolated in your laboratory that had been applied in pharmaceutical or in human recently.
Yes, I'm that, but in our laboratory, we can evaluate some compound using a mice level. So if we use the dog, of course, human, we can collaborate with pharmaceutical companies.
Jadi jawabannya levelnya itu baru pada level anjing, belum sampai ke manusia. Nah, yang tadi yang disampaikan oleh beliau, kalau harus level manusia, berarti harus bekerja sama dengan perusahaan farmasi. Dan itu yang butuh funding sekitar 10 juta yen. 1 yen itu kurang lebih sekitar 100 ya. Itu kurang lebih sekitar 1 miliar lagi gitu ya, untuk bisa mengaplikasikan itu ke sana. Cukup? Penanya? Oke. Ada ini
Okay, thank you very much for all participants in our discussion this afternoon. And to Ari Sensei, arigato, thank you very much for your presentation, and it's nice to meet you in this session. And we give applause to Ari Sensei.
Thank you. Terima kasih.
Oke, terima kasih kembali, Ari Sensei. Oke, I think enough this session, and thank you very much to all participants, and we will to take picture in this session. So all participants, please open your camera. Yang ambil siapa? Operator siapa yang ambil nih? Yang Oke. Operator siap? Oke. We will take picture. 1, 2, 3. The next slide. 1, 2, 3. The last session. 1, 2, 3. Okay, thank you very much. I think enough for our inbound lecture today. And thank you to Professor Masayushi Arai, Mr. Hesatria, and Pak Mulyono, and all lecturer in this room. Okay, thank you very much.
Thank you very much, Professor.
And we close this session. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon.
Yeah. See you. Bye bye. See you again. Okay.
The speaker in camera 1 is off, so we could not hear. Maybe operator can activate it, the speaker from camera 1.
Itu perkuliahan ini mengingat apa? Kita sudah referensi event yang seperti ini adalah event yang menunjukkan suatu event yang prestisius ya. Kita bisa berkolaborasi, Pak Andi, Pak John sudah memulai knowledge yang sedemikian banyak dari para pembicara, baik kemarin yang dari Malaysia, kemudian hari ini yang dari Jepang, dan masih satu lagi ya, satu lagi dari Malaysia ya, dari Malaya University. Malaya University itu masuk ranking 67 dunia. Ya, jadi apa yang menjadi speaker dalam kegiatan inbound lecture itu sangat luar biasa dipersiapkan oleh tim yang oleh jurusan. Harapannya apa? Itu membuka cakrawala wawasan Anda semuanya, khususnya 2020 yang sekarang sebagai mahasiswa yang menjalani riset. Makanya itu kami wajibkan di sela-sela Anda melakukan kegiatan riset ataupun persiapan yang lainnya. Ya. Harapan kami apa? Nilai tambah itu akan memperluas Anda. Barangkali yang tadinya nggak pengen sekolah, tadi Prof. Are menyampaikan, oh ada kesempatan bisa sekolah begini dan seterusnya. Ada yang interest bidangnya mau meneliti yang aplikasi kemana, itu sangat terbuka lebar. Ya. Wawasan inilah yang diharapkan Anda peroleh, ya, di samping Anda semuanya akan mendapatkan pengetahuan yang sangat luar biasa dari berbagai macam riset yang Prof. Are tentunya profesor-profesor narasumber yang berbicara itu dia meng-hire banyak mahasiswa dari level S1 sampai S3 ya. Sebagai closing, masih ada satu kesempatan lecture lagi di tahun ini ya, tahun depan nanti akan kita arrange lagi. Bapak tanpa perlu kemudian memberikan announce lagi yang dengan ekspresi yang tidak menyenangkan ya. Jadi ketika nanti ada kuliah lagi jam 10, Bapak minta jam 9.30 semuanya sudah hadir. Ya, itu salah satu etik kita menghargai mendudukkan seorang profesional, seorang akademisi dari perguruan tinggi world class university untuk kita hadir.